Hey everyone, how you doing? It's Clara and I'm back with another one of my science chats where I talk to people who work in STEM fields, so science, technology, engineering, mathematics and medical science, about their research background, about what they're doing now, and also about making science welcoming for everyone, more representative of the population. And today I'm talking to Dr Lilian Hunt, who works for EDIS, which is an independent charity in medical science, which is founded by the Wellcome Trust, GlaxoSmithKline, and the Francis Crick Institute, as well as a whole bunch of other medical science institutes working on making medical science more improving the equity in medical science. So it was great to be able to chat to her. I also was able to chat to her a little bit about the ethics of animal testing and stuff like that, which wasn't intended, but it was where the conversation went. It was quite a long conversation, but in a good way. And so, yeah, I had really good fun. And it's also really nice to have someone on my channel that I think so far all my guests, like Chris Jackson and and Andrew, everyone's been working in research institutes in universities, and so this was a good opportunity to talk to someone who done research but then sort of moved slightly outside of that, so we're getting a different perspective on science careers, really, as we will with our next guest as well, but more of that later. So yeah, without further ado, let's go and chat to Lily. Hi Lily, how are you doing?
Hey, I'm good, thank you. How are you?
I'm good, thanks. Yeah, so technical problems aside with internets and crashing TVs and microphones not being plugged in, we're doing all right. I think we're doing good.
Yeah, got it covered.
Yeah, technical. Yay. We build the equipment, we don't have to know how to use it, I think. So
a bit of practice.
Yeah. Nothing. I'm worried about a conference that's coming up where technically we're in charge and I don't think we've got tech. I'm like, oh. Anyway. So for the audience at home, can you introduce yourself? Tell us a little bit about yourself and who you are and what you do.
Yeah, I feel like I should have prepared this bit a little bit better, knowing that you probably would have asked me who I am and what I do, but here we go. So I'm Lily Hunt. I work at the Wellcome Trust at the moment. I'm the EDIS program lead, so that's the Equality, Diversity and Inclusion in Science and Health program, which is actually a coalition of organisations across science and health research. And then we've got about 18 member organisations branching funders, society, research institutes, different charities, things like that. So that's what I lead at the moment. My background is I did a PhD in genetics. It was at the National Institute for Medical Research, which transitioned into the Francis Crick Institute during my time. So that's kind of my background in terms of who else I am. I mean, it's very hard to tell these days with lockdown what my hobbies are. So I guess staying in the flat is sort of my main hobby. I don't know, like. Previously, I'd be like going out, having fun. No, I like the living room.
Yeah, massive interest in cleaning the kitchen and things like that. Yeah, we've all been there. It's a weird old time. It doesn't help when you. Yeah. Well, yeah. Let's not. I was just about to try and do a riff on cleaning the kitchen, but I really don't need to. I think we've all cleaned kitchens before.
Sure, yeah. We've already crashed the conversation. Brilliant. I'm excited for the rest of this.
Already crashed Clara. That's easily done though. So I need to reboot. So yeah, so you work at the Francis Crick Institute, which is in London, and which I have been to once, which is kind of exciting. Or twice, I think. I don't know. I've either been there once or twice, but you're there, or you normally would be there quite a lot, I suppose.
Well, I mean, so I did my PhD there and finished that in 2017. Gosh, maybe a little bit earlier than that. Hard to tell. What year are we in now?
Yeah.
But then I've been at the Wellcome Trust ever since. But the Francis Crick Institute is one of the founding members of EDIS, so I do get to communicate with them there quite a lot and go over and have meetings here and there as well. But most of the time, lockdown aside, based in Wellcome Trust on the Euston Road, which is a lovely place to work.
Oh, okay, cool. This yeah, I didn't, I don't know why I didn't realize that. And so yeah, you did your PhD in, well, what is now the Crick. So what was that on? What was the sort of topic? What was it you were looking at?
Yeah, so I was working specifically on conserved non-coding elements within genome. So this is something that my boss, Greg Elgar, my supervisor, he was really involved in the sequencing of the pufferfish genome, which is one of the most compact genomes that exists, basically. So the idea is that there's a lot less of what we used to call junk DNA in a pufferfish genome. So when you compare the pufferfish genome to the human genome, and actually vertebrate genomes in general, you obviously have a lot of genes that are homologous to each other and lots of replication there in terms of what you'd expect. Pretty much most vertebrate organisms run off essentially the same set of genes. But then outside of the genes in the non-coding regions, he found loads of chunks of sequence that were basically almost identical that weren't genes. And the idea was like, okay, so we've had millions and millions of years of evolution between the ancestors of a pufferfish and a human, and yet we have all of these chunks of DNA. I think it's something like 6,000. God, this is tracking back a little bit. 6,000 of chunks or so that are almost identical, that have survived all of that evolution, all of that potential mutation by chance, despite the fact that it isn't the same selective pressure that genes have. So, if you have a mutation in a gene, that could mess up the protein function. You're quite clearly, that's not going to happen too much in the population, or at least you'd hope it wouldn't happen too much, because then you'd have faulty proteins going on. Whereas these non-coding elements, they're not coding the proteins, but they're still exactly the same almost. And then when we looked across all vertebrate organisms, they kind of existed in exactly the same way in all vertebrate species. So this was an area of interest, and it was kind of back when we were just moving out of the idea of all of the non-coding genome was junk. Was what, when I started, I think it was maybe my A levels, that was kind of the idea that all of this stuff is just extra stuff that's just been kept. We've not got rid of it because the human genome isn't evolving in a directional manner or in an efficient manner necessarily, but it's there. God knows where some of it comes from. Is it viruses, things like that? All of this sort of stuff was being speculated when I did my A levels. By the time I was through to PhD, we're like, hold on, some of this could actually be interesting and could actually be useful and could have a reason why it's still there. So yeah, having that evidence of sort of these regions surviving, sort of, I guess, evolution tells us that maybe they are important for something. So then the inferring section was like, okay, if all of this stuff is the same in vertebrates, what is the same in vertebrates? And it's the whole thing. It's the head, the spinal cord, the neural tube, that sort of stuff in early development specifically. You look at, albeit slightly skewed drawings of embryos where vertebrate embryos were drawn next to each other and made to look very similar. They are actually a bit similar in vertebrates, that sort of, there's a couple of stages that look pretty similar. But most importantly, it's like the process of development, which includes the neural tube, the spinal cord, the brain, the sort of that section that makes a vertebrate is pretty similar. So my boss's previous work was around looking at whether or not these regions were gene enhancers or instructions for the genes, basically. So the way I like to explain it is all of your genes are the ingredients to make a cake, okay? Which is great. Got all the ingredients out in front of you. But can you imagine I just gave you all the ingredients and didn't tell you when to put what into where, how much, or when to take it out of the oven? You wouldn't really be able to make a cake, let alone would everyone be able to make cakes in the same sort of way. And that's kind of like making a human. You got all your genes, you've got all your ingredients, but you need to know how to put them together. So we're looking at these elements, and what you can do is you can take out these elements of the human genome, these conserved non-coding elements, stick them in front. This is really crass language here. Stick them in front of a jellyfish gene, GFP, green fluorescent protein. and then inject that into a zebrafish embryo at sort of the 1-cell stage, gets taken up by the zebrafish cells, and then over the course of the next 24 to pushing to sort of 72 hours, you can visualize the zebrafish under a microscope and basically look for the areas that light up green that tell you that that section of human DNA that's conserved on coding element is kind of instructing genes to switch on in that area.
Wow.
So I've got loads of great pictures of zebrafish embryos sort of about 24, 48 hours old with just like little green light up bits in their brains, basically.
An interesting
that's really
thing
cool
to have.
yeah. I mean, more interesting is the fact that I think one time I went to my supervisor's office and he just pulled out a pufferfish dried skin from his like drawer and was like, "Look at this," and I'm like, "Why have you got that? Never mind." So that's kind of like the enhancer side of things. So that's kind of like a load of work that came before my PhD, really. And then my work was then taking groups of people who had mutations in those genes that were either related to so groups of individuals that were either sort of patient groups, so they all had like the same sort of developmental disorder, for example, or groups of people who had something interesting biologically going on, basically, which we think might have been to do with sort of development differences, but we weren't entirely sure where. So I sequenced all of their conserved non-coding elements, basically, and looked for mutations that were different amongst those groups compared to those expected in the general population. So then started experimenting with whether or not, if I injected those enhancer elements, so those conserved non-coding elements, in their sort of wild type form, looked at where they lit up versus in their mutated form, and saw if there was any difference into where they lit up in the zebrafish embryos as well, to see whether we could figure out anything about the language of these conserved non-coding elements, and basically, do mutations matter in them? Do single point mutations matter in the same way? Like, do they do in genes? Is there any information we can gather from that could say, like multiple mutations across multiple enhancers could lead to sort of disorders and things like that? And it was a really interesting project, more questions than answers at the end of it, like I think most PhDs have. And I did a really cool study actually with a group. It was a group in Denmark, and it was a obesity study. They had a really well sort of like data managed group of individuals who were sort of in the extreme end of obesity, who they tracked from childhood basically as well, so we could look at historical data. And basically we used that information to look at, specifically, there was like about 2 million bases worth of DNA that had conserved non-coding elements in it, that we were looking at for mutations there as well. And that kind of inferred to us actually a bit about the mutations in some of these areas potentially being linked to a specific set of genes that were actually more involved in development than anything else. So this is the IRX cluster, whereas previously, like, research had been looking at maybe like the FTO genes, because that was where the mutation directly was linked. So that idea of like these long range enhancers, mutations in them, plus at the same time some stuff around topologically associating domains was coming out. So this is kind of how DNA ties itself in knots, so that two bits that are really far away are actually really close in space. And that tells us that, you can get a gene over here and an enhancer over here. And when we look at it linearly, you're like, oh, they've got nothing to do with each other. But then we started mapping how DNA was like coiling up in terms of like knots and stuff, and we realized that those two bits are actually really close to each other, and therefore likely to influence each other. So all of that kind of was happening at the same time, which was really cool to see. So it's really started sort of like, it was really nice to be at that sort of point where people were really sort of turning all of it. So we'd had the idea of this drug, realized it was important, and now we're starting to realize what some of this stuff was actually doing. I feel a bit out of touch with the research now, but it definitely felt like we were at this point where we're starting, like, really early stages, starting to understand a bit more around like language of like the DNA in these sort of enhancer elements, and sort of this idea of the 3D structure of DNA being so important within all of this. So yeah, it's a really long-winded explanation of like everything I've done in research.
Well, that's because you've done a lot in research, and it's great because I don't know any of this, so I hear terms like junk DNA and I've got an idea of what it is, and I've been hearing that it's not good to call it junk DNA because actually we kind of need some of it. But
Yeah.
it's really good as a layperson in this regard to sort of hear more about how it's working together, and even the shape and how it orientates to around each other is just as important as everything else. And I think it was important to hear you. Well, interesting to hear you talking about how like viruses could leave their marks on DNA potentially, or I don't know if that's what you were actually saying. That's what I
I mean,
picked up.
there's definitely thoughts around that. There's definitely thoughts about like archaic, like for example, like archaic like bacteria back in the day, like inserting itself into the human genome, and that's actually the way that we have jumping genes. I think is the best way of describing it. We've
Okay.
got bits of DNA that can actually like move around and stuff like that. That doesn't come initially from humans. That comes from external, and it goes into our DNA sort of at some point. And we actually now we've utilized that and use it for when we're trying to do experiments, for example, with DNA as well, which I think is I like that whole sort of cycle of like there are bits that we can potentially explain by DNA like insertions from other organisms, but then also to then sort of recognize some of that and then utilize it to our advantage within research, I think is also like a really neat way of like looking at this sort of stuff. So, even the stuff that back in the day you might have been like, oh, it's useless, like it's becoming useful in different ways.
That's really interesting. I know this will sound really probably really obscure, but you're talking about zebrafish, and I feel like I've heard a number of talk on zebrafish. Like a zebrafish, what is it about zebrafish?
What is it? What isn't it about it? Zebrafish are great. So zebrafish, the biggest thing is that you can actually gosh, I've literally lost off the top of my head what it is we add to
Oh,
zebrafish. That's
sorry.
just awful. Yeah. You can add something to zebrafish embryos, and they continue to develop transparently for a number of days. So really great for microscopes, microscope imaging, and also anything involving like fluorescent proteins and things like that as well, because you've got these completely sort of transparent or translucent embryos. They have these little round yolks that they kind of like develop around, which actually means that you can actually create these really cool petri dishes with like dips in. You can actually just stick a zebrafish embryo kind of like so it's stuck in the dip, stays on its side, and microscope image that across like 3 days, and you'll be able to then create like these beautiful time lapses of embryo development from one cell stage all the way through like 3 days old, just before they're sort of like ready to go off swimming in their little dishes a little bit more and stuff. So they're really great visually to use for sure. Other cool stuff, they have like ability to like regenerate cells within like their heart and stuff like that. So that's really cool. You can start to see things around like blood circulation as well at some points and things like that. And obviously there's a lot of like homology and similarities in terms of their development, other vertebrates development, human vertebrate development. So yeah, there's loads of uses of zebrafish, and obviously they're quite easy to look after, and you can have lots and lots of them, and you can breed them very quickly and very easily, and you can tell the difference between the males and the females, and there's loads of like just handy things with them. I think one of the things that I found quite interesting was the amount of time it took to kind of realize that if you sing to them or speak to them, they seem to breed better. Now, I don't know if this is real, but it's always worked for us. Like, I remember in my lab being like, oh, the zebrafish aren't laying eggs. And one of my lab mates would be like, have you spoken to them recently? And I'm like, what are you on about? And then go down, have a chat with them in the aquatics facility. And then the next day, they're fantastic. So, I mean, that's a study of n=1, but I think it works.
Oh, wow, I'm imagining sort of James Brown type songs going on here.
Well, that's the thing. Like when you breed them and stuff, you have to for egg collection. Firstly, they lay hundreds of eggs at a time. They can get anything from sort of 100 to 200 for like spawning. They do it quite quickly as well, and they'll only do this as well when they're in sort of shallower water. So while they're in the big tanks, you're absolutely fine. They're not breeding or anything like that. So you just take them out sort of in their little male female pairs. Get like a little tank that's maybe sort of like that deep or so, with a divider in the middle, male on one side, female on the other. Sort of make sure they're seeing each other, very excited about what's about to happen and things. And then you like let it happen. And then sort of
Wow.
within sort of an hour, you've got like a whole tray of eggs at the bottom, which have all been fertilized pretty much all the time, you know? So it's like they're trying to help us, let's say.
Sounds like an early 2000s Channel 4 TV show. I
Well,
think,
Joe, because when we moved into the crick, we had this new thing. I can't even remember what the actual name of it was called. I think we called it the ice spawn, maybe. I'm not entirely sure, but definitely it was called the ice spawn. But so previously we were doing, one fish, one fish, one fish, one fish, maybe sometimes two fish, one fish, per little menage a trois if they're lucky. Um, but the ice spawn, I think, was like you can put like maybe like 50 fish in this thing, and then like kind of just let them go, and then you just get like thousands of eggs coming through, and like it's just one of those ones where you like stepped up the production. And in your head, you're going, "This is great for my research, and I have like all of these eggs that I can use and things." Then also you're like, "Have I just made a fish orgy?" Yeah, I have. I have just made. Yeah, that's what I've just done. Yeah. Mm. We're going to need to come up with a better technological word for that to go into my thesis.
yeah, you'll have to call it Love Island Tank or something. Uh, and when you were just talking then, I realised that I think I'd heard a lot. I think there must be a group at Oxford that do a lot of work on the regeneration of the hearts of zebrafish. And I think am I right that there's like two batches of zebrafish that have developed separately so that they're pretty much identical, but one group can heal and one group can't? And I don't know. You might not know that. I realise that might not be your subject, and I just have happened to hear a couple of talks on
Yeah,
it.
it's slightly further away from my subject for sure. But what I do remember is that when I had my interview for my PhD, we were given a kind of, I think maybe 4 papers or something like that, and you've got to pick one that you could then sort of present on. And I do remember that the paper that I picked was on the zebrafish heart development, and they'd just done the rainbow assays, I think it was called, which was basically where you could basically individually like tag cells in development with different colors by using like mixtures of colors of different genes and proteins, and basically create these incredible, beautiful rainbow sort of organs after XYZ levels of stages of development, because the cells, each of the individual cells that was tagged was able to then, as it sort of multiplied and went across times of development, they'd be that color. So you'd see where the origins of those cells would end up, and you could see these beautiful sort of multicolored sort of hearts where you could see, okay, this section here was from this cell here, and this section here was from this cell here. So that sort of stuff was happening at the time where I started my PhD. I remember just thinking that that imaging and the stuff that was going on there just felt so beautiful and like artistic, as well as like obviously incredibly useful and giving us all of this information. And I think I was presenting the paper and they were sort of asking, what are your thoughts on the paper? I was like, my thoughts are they came up with this method and just wanted to show it in a really beautiful way. And they found out some interesting things about sort of like what developed first and what didn't. But realistically, that wasn't really the purpose. The purpose was like the technique and the imaging and the colors that came off the back of it, and being able to prove that you could do this. So things have moved on a lot since then for sure. But like even back then, I just knew how special it was to work with sort of imaging in development, and how important that was.
Wow, that's so cool. Yeah, and I love that. Well, I mean, that's it. I think this is a lot. I've talked with a few other people and proving the concepts of processes, proving that you can do something or that there's better ways of imaging, or I mean, that's a big part of it. Seems like it comes up in most of these talks, to be honest, is like, how do we image in the best way, whether it's on the nanoscale or whether it's on a much larger sort of scale, in planets and stuff like that. It's
Yeah, for
interesting.
sure. I mean, like, we're humans, we're visual people, we're visual creatures. As much as not everyone maybe have the same sort of visual abilities, by nature of who we are, everything at some point gets turned into a photo, a graph, an image, or something like that. So most of the research that we do, in some way, shape, or form, has to make it into that form at the end of it. Even when we're looking at genetics, even when we're looking at DNA, I there are genetic visualizations that happen. The genome visualizations are a thing, and you end up with a graph on your screen with all of the letters, all the A's, C's, G's, and T's, in their different colors, because they know that that's how we work. You know, you never show the molecules, themselves. You show the letters that represent them, and you turn them into different colors. And you look at, for example, like as a really crude example, the path that is in Cambridge that goes to like the Sanger Institute and across from the Sanger Institute to Cambridge is like a DNA path, and that's like coloured blocks in different orders that represent a section of human DNA. Like it's all about that visual medium as well at the end of it as well.
I didn't know that. I've not actually spent that much time in Cambridge.
Oh.
So spend all the time at the enemy. Mostly it's just so hard to get to from Oxford. I think if there was a way of getting there quickly and easily, I'd have been there an awful lot more than I am. But we've got to go through London, and you're talking 4 hours each way, which is quite remarkable, really. One thing that is interesting, so I work with kind of inanimate things. I work with vacuums and metals, or maybe plastics and ceramics. But all this work, you're talking about it, and I think probably once or twice I winced a little bit, because you're talking about embryos in petri dishes and injecting things, and I think I realised that we need to do some of these experiments. But also, I've been a vegetarian for 26 years, and I'm kind of very much like, save the animals, but and it's really difficult. I realise that's probably an awful lot of big topic on ethics, but what are your thoughts on that?
It's really important. I'm actually eat vegan now, and I have been at least pescatarian my entire life. So the work that I've had to come up with this for myself, like figuring out how do I take my personal ethics around the consuming of animals, and how do I rectify that against the fact that I'm doing this medical research, which will inevitably involve animals at some point. And I knew this sort of kind of from probably GCSE biology, when I was really like, okay, this is the path I want to take, that I was going to have to figure out how those two sides of me sit together in a acceptable way. And there was like a huge internal debate as to like, am I justifying something that I actually don't believe in order to follow this other thing for selfish reasons? Am I not? All of these conversations happened in my head. And we also had some really good ethics lectures. So I went to, my undergrad was at King's College London. We had some really good undergrad ethics lectures where they genuinely gave the platform, like the lecture platform, to people on complete opposite sides with zero framing of these lectures. So this was for like animal research, for example. I'm trying to remember what the other ones were, but like that one really stuck out to me, where it was like, here you are, here is someone who is somewhat of an animal rights activist and a scientist of some form who doesn't use animals at all. Here's another researcher who does use animals, and there was no framing. They gave us no context or anything like that, because it was very much like, look, here's two sides, make up your own mind. Like, we're not going to sway you on anything. You need to make this choice yourself where you feel comfortable. But I really I thank my university a lot for giving us that side of things. And what it led me to think was basically I needed to come up with my own line in the sand for various things. I have my personal ethics in terms of how I conduct myself. I don't eat meat. I don't eat fish. I don't eat dairy products. That's my personal consumption. Now the question is then, okay, how does this relate to medical and health research? And it was kind of understanding what's necessary in order to progress not just human health but also animal health as well. And what is the way to relieve the most amount of suffering in various ways, and also therefore what are my limits in terms of where do I draw the line in terms of what animals I work with? Where do I draw the line in terms of like the age of the animals that I work with and things like that? And that's where it kind of came to that point where it's like, okay, I saw mice being killed during my undergrad as part of research, and it was kind of one of those ones where it's like, oh, do I think I could ever actually do that myself? And to be honest, I don't think I can. And because of that reason, I would probably not choose to do research with mice or with mammals of that type and higher. partly because I think you need to take responsibility for your own actions, and I couldn't also therefore legitimately feel comfortable doing research on animals that I've got someone else to kill. Like that just doesn't, that kind of feeds into my same psyche when it comes to like not eating meat myself as well. Like I could never kill an animal, beside the fact that I think there's other reasons as well, but like I could never kill that animal. So therefore I'm not about to pay a supermarket to go and kill it, chop it up and put it into nice packaging so I don't have to deal with that consequence. So that's kind of where that kind of line came for me in terms of what animals I was willing to work with. And then when it came sort of like around the fish side of things, yes, I think there is still issues in terms of sort of animal husbandry that need to make sure they're always at the forefront of everything. Animals, if we are going to be using them within health research, within medical research, there is so much about, making sure that that is an appropriate environment, that they are well looked after, that the people looking after them and people using them in experiments respect the fact that that is still life that you're looking after there. The thing around with embryos, though, I think is where I have a very different viewpoint, and that's because I'm very much pro-choice, and in terms of human side of things, and in terms of animals as well, that obviously comes through into the same concept of like, where do I draw the line at where life starts, like that. And for me, that's a very different sort of a different conversation entirely to consumption and like adult animal life and things like that. So I think that there's a lot of complex thoughts around that. But first and foremost, I will say that, there are some things that if we want to progress in certain parts of health and medical research, we can't do it without the use of animals at the moment. It would be great. And I think there are a lot of people out there who are working really hard to get us to a position where we can start replacing animal models with cultured cells, cultured organs and things like that, which was going to be, it's phenomenal when it happens. The problem is we're not there yet. And that's why it's quite hard, because we can see it. We can see this opportunity to remove so much, animal death from our research, but we're not there yet. And it's just making sure that we keep a realistic track of where we get to. I mean, I think it's the same when you think about actually to get to that point as well. There needs to be animal experiments as well that leads to it. So it's this constant cycle of sort of weighing things up. And my opinion won't be the same as the next person's. And there'll be other people that will think I'm wrong and other people that think I'm right. The idea, I guess, is to make sure that we have a well thought out scientific consensus and an understanding of ethics and ethos behind things. That means that we work in the best way for sort of the general sector, and to very much take everyone's thoughts into account when it comes to this. I think that's really important. This can't be led by like one person's thought somewhere saying this is how we do things. It's got to be a continuous conversation that is really reflective and agile in response to research that's being done. I guess the only real thing at the moment as well is that for medicines to be given to humans, basically, there needs to be some animal trials before that. And this is, I'm very clear, by the way, this is medicine and this is research, medical and health research. This is not cosmetics. That is a completely different thing. It's very much like thinking about this box specifically. And I think there's loads of great work that can be done on this. And I think the idea of this sort of like reduction of animals, and refinement of experiments and things like that, I think is really, and replacement, wherever possible, I think is, that ethos still needs to remain, and I think it really does. I think people. I remember being in primary school and we had debates about animal testing. And I remember going to the library and getting a book out and looking through at the pictures of bunnies with sewn up eyes and things like that. And like it's a very different world to what we're living in now and what we're talking about now. This is very specific. I mean, there's stuff that I think one quote that really stuck with me actually from those lectures when I was at King's was that you can't make a cell cough in culture. Now, a cough doesn't feel like much maybe to you or I, but there are people out there with debilitating coughs that mean that they crack their lungs their ribs every time that they have a coughing fit. How do you weigh up those things on someone's conscience of like, oh, we're not going to help you, or versus actually we can. And then once we've done that, we no longer need to use those animals for that process anymore. It's all this sort of stuff. Yeah.
I think it's the same with everything. You've got to have all the information, know what you're doing, and make your decisions. And it's good that King's gave you those lectures to allow, they weren't trying to inform you or change your mind in any way. They gave you the information. I think it's good that you've got your lines and you know where it is, and you can, you've taken that internally. And like you say, it sounds like for you there's a big difference between animal testing and medicine, and I agree. I completely agree. And I realise that we do, unfortunately, we are still in the point where we do need to test on animals as much as I don't want to. Like you say, we need to be able to sort of work on medicines for people that have got debilitating illnesses. And
Yeah, for sure. I mean, you've got so many medicines out there in the world today that are really, they save lives. They help people, live on a day-to-day basis. You think about like an asthma pump, for example. Like without animal research as part of this, we wouldn't have some of those things. And it's you've got to remember that as well. I think the biggest thing I will say is that, every scientist who's going to be working maybe with animals needs to have this conversation with themselves, basically. And I think all of undergrads, across the country should have this in their lecture series, maybe even, in their like tutorials or something like that, to say, okay, really think, how do you feel about this? How do you feel about using the animals yourself? Animals that you could be using. There are animals, things you can use all sorts of animals in research, and sometimes you have to, and sometimes you don't. And it's this idea, you're going to have to face up to it at some point. You better do it early, because otherwise, you could end up, far too deep in research and realizing that actually there's something that you need to do that you physically don't see yourself being able to do. And also this conversation, I think you have with yourself kind of continually as well, and making sure that you are continually comfortable with where you're drawing lines and boundaries, because people's ethics do change. People's moral lines, they will always evolve. There's learning that's always being done, hopefully by everyone, to inform their choices. So you have to keep that going. And I also don't think, I would never look down on someone who's using animals in research in a way that I wouldn't either, because if they've had that conversation with themselves and with the research that they need to do and things like that, I think it's it's very different for each individual. So I think it's very much trying to think about this non-judgmental place, putting the animals' needs first, as much as possible. It's making sure that if we are going to be using animals in research, we're honest about it as well, especially to the public. The open reporting of how many animals that each institute is using is really important. There's no point shying away from this conversation. You have research institutes with flaws of animals that are being used in research. If we hide it, it makes us look even worse, and it makes us, it makes it seem like we're hiding poor maintenance, poor animal husbandry or things like that. It's not like that. I think all the animal research units, they've got vets that continually. The idea is to look after the animals, and it's not an awful place, I think, is the best way to describe it. It's making sure that we are continually improving the way that we use animals in research for their benefit and for ours.
Almost sounds like I'm pretty absolutely sure, in fact, that some of those animals are actually probably getting treated better than people's pets.
Yes.
Which, yeah.
And I think,
my view on that is that not everyone should have pets, but
agreed. And I think, bearing in mind neither of us eat meat, I think the idea that someone might slate my use of animals in research and go and have a sausage at home, I found it very difficult to have a sort of debate about something like that. So there's a whole section there.
No, I completely agree. I think it's good that, I think it's good that King's had those conversations. And I like the fact that, hopefully that's part of the culture now, that it isn't just animals are inferior, that, universities are actually changing the way they work and their ethics and their principles, and being more open about these things. Like you say, not hiding anything and not pretending and not fooling themselves. And if that is part of the culture, and like you say, as whenever it's possible, moving away from animal testing, then it's more of a positive. It's still rather it didn't happen, but you can understand why.
And that's okay, because I think that's still, that's the end goal. If you aim towards that, which is like, we can move away from using animals in research entirely, and you do it with, I always like to talk about intention, just in general. This is like in life and science and everything like that. If someone has good intentions, to genuinely get to that point and to improve, then I think that is really the thing that is the most important. The intention isn't to harm animals in terms of but for no reason really. The intention is to move away from using animals entirely. So let's keep going down that path. Let's make sure that we are funding that research as much as we're funding research where we are using animals. Things like that. I think that's really important. And again, the intention around like universities or research institutes, reporting and stuff. There's no, like I said, the intention isn't to hide. The intention is to be honest. You have to be able to look at yourself and say, okay, if everything of me like around this was exposed, would I feel comfortable? If the answer is no, you're not doing it right, and you need to look at your own practice. Whereas if the answer is that yes, if I was completely honest about everything that I've done in regards to animal research, I would feel comfortable with people knowing that. And I think that's really important.
Yeah, no, thanks for that. I realise wasn't necessarily what I thought we'd be talking about, and possibly neither did you. But it's something that even just as a bystander, it's something I struggle with. The point of I know that we need it, but I also wish that we didn't. And so, in some ways, it's kind of nice to know that at least relatively they're being treated well and trying to move away from it, and people are not just sort of using these animals like someone picking up a slab of meat in the supermarket.
Yeah, exactly. It's a completely different world. I remember as well. I remember when there was a piece of research into the housing of mice and how to make them less stressed, basically within their own within their little, I want to say cages, but like cages brings up an image which is not the same as what happened, like tanks, more like giant hamster tanks type thing.
Yeah.
Anyway, so one of the things is like actually the mice being observed was somewhat stressful sometimes, and they very much needed like their own space. But obviously, as a researcher, you needed to be able to see them in order to check up on their welfare and things like that. So you look out for things like have they chewed their fur and things like that, and that that's a sign of stress. So you want to make sure that they're not in stressful positions. Making sure that they are not being used in experiments, for example, or being handled. If they show any signs of stress, you want them to be chilled and relaxed. And so there was a little sort of perspex like hut that got put into the mouse cages. There was a specific type of like red plastic which had like particular refractive properties, which basically meant when the mouse was inside, it was like a little dark hole of like comfort, like a little cave for it. But we could still, as researchers, see in, so we could see that they're sleeping well and having like still relaxed and things like that, the outside, without having to disturb them at all. And it's like little things like that, like the small practices that they're really cheap to make, and they were like rolled out almost instantly across sort of like all of the cages and things like that. And you're like, okay, this is really cool that we are still putting time and effort and energy into this, and it's really important that we do this. And I think there's some research out recently around how to handle mice as well to make sure you reduce stress. Like the comparison of whether you're picking up sort of with the end of the tail that's like close to the bum, basically. If you use that as sort of a main sort of mechanism of picking them up versus if you pick them up using, I want to say in my head, I think I've probably read this slightly wrong, but like the version of like cardboard, and like scooping them,
Okay, yeah.
and like the differences that can make to their stress levels and things like that. So continually improving, even handling, I think, is also really important. And I know that like the NC3Rs, the research and the sort of publishing of this when it comes through is really important to the whole field.
That's kind of interesting. It's really good that they're sort of looking at and putting active time and resources into looking at this. And I don't know, maybe this is the cynic in the back of my head, but are we doing the same with our researchers? I mean, I don't know what the field's like, but
I mean, that is all the other fields. Segue.
Yeah. Thank you.
Yeah. Are we matching the research that we're putting into reducing stress of mice with that of reducing stress of researchers? Probably not. Probably not. Especially recently, probably been an incredibly stressful time for everyone. And yeah, so much needs to be put into that. I was actually just had an EDIS meeting this morning, and there's a number of our members that have been involved in surveys and studies at the moment, speaking to researchers, try and find out really how much this pandemic and this lockdown has impacted mental health, well-being, career prospects, how they feel about the research that they're doing right now, what they think the impact might be, and things like that. And hopefully we'll be able to pull all of that together, because we have such a broad variety of firstly members, but also the types of information they've been gathering. And we want to pull that together because the insights are incredible in terms of like understanding what's the immediate impact versus what's the sort of medium term impact and the longer term impacts that people are seeing that are going to happen. But yeah, I think that this is the time where we really have seen already inequity in terms of resilience, was already there. And now we've had this huge stressor situation, and it's all come out basically. And I think that's a prime example of why so much work needed to be done before all of this happened. But now, if we don't do something about it, we're almost in crisis mode.
That's kind of interesting. Do you think? I mean, I sort of believe that things like, and I realize this is a different topic, but things like Trump in America are inevitable, because if things are just slowly declining or we're becoming ambivalent about things, then of course things have got to get a lot worse for us to take note. Do you think that in academia, in the academia sort of climate, do you think that this is that sort of crisis that could actually, it means that we can look at how we're doing things and how we're treating our researchers and academics? And do you think this could be a positive, could potentially have positive? I realize you can't see the future.
Yeah, I mean, I think one thing I will say in terms of things like around, for example, Trump in America and those types of things, is they're actually, I don't think there's necessarily ambivalence that has sort of led to that. It's actually been quite a lot of polarization, I think, that's been going on in terms of sort of like the two camps of, like, for example, left and right wing politics. Now, I think we're in a position now within research where we're thinking about, there's a lot more people talking about changing research culture and things like that at the moment. And obviously with that, I do worry that there's a concern that we could end up polarizing the research sector as well into those who want to stick to the current system and don't see what's wrong with it, for example, or don't you think that this is the right way to do research, versus those who really want to move to a completely different version of how we structure things. I don't think that's healthy if we were to completely sort of split into these two polar camps. What we really need to do is create this sort of like overarching movement of change into a better version of what's going on. I will say I'm very much stuck in sort of that camp of sort of things need to change. Things have needed to change for a while. And this does feel like a moment in time where we've got clear evidence of inequality. We've got clear evidence of the impact of stress situations and sudden catastrophes, essentially, on different people. And we've got enough people now really sort of like wanting to change, I think it feels like. Now, I could be looking at this with rose-coloured lenses of sort of D&I practitioners for the past sort of 30 years have seen this sort of cycle happen, all the time. But we haven't had a global pandemic in that time, so that's why it does feel different. And the thing now is to really harness that and sort of figure out how to move forward. I think there was, like I said, they had this EDIS meeting this morning, and there was this exceptional sort of like moment where people were just basically like, you know what, it's time to rip up the rule book. It's time to like design something from scratch and see where that lands, because this feels like the time where we could actually get away with proposing something completely different. And I really was sort of like, I've suggested this a few times, like to different people throughout the past however many years, and each time it's been like, oh no, we can't get rid of the new system until we have a new version. Like, can't get rid of the old system until we have a new system in place. Whereas now it's like, all right, so let's start on that new system. Let's figure out what that could look like and see where we get to. And I'm sort of like, oh, so all it takes is a slight global pandemic to get people to really like want to change everything completely dramatically. Fantastic.
Or we need hundreds of thousands of deaths and we're fine.
Oh gosh, don't talk. Oh wow, really bringing it home there, isn't it? God, yeah, no, it is upsetting that it's taken something as huge as this for things to change. In the same way, for example, like if you think about the resurgence and surgence of the Black Lives Matter movement over the spring and summer, it's awful that it's taken highlighting police violence and murder in the States of Black people to make people do anti-racist learning in their day-to-day. It's awful that it's taken that, but it has had an impact, and it's really difficult to then be like, okay, that's happened, which is awful, and I would love to be able to, people want to say like this shouldn't have happened because of this. It should have been happening already. Unfortunately, we can't go back in time, so it's like, okay, let's try and harness this moment and make sure this is happening. And there are organizations that have obviously made outwardly public statements around racism in academia, in research, in their own institutions and organizations, and I commend those who spoke frankly and honestly about their own part in it as well. Now it's just making sure that those organizations are held accountable to things that they've promised. Have they said that they're going to change their ways and do things about it? Yes. So therefore, they need to do those things, and we need to go back to them in X amount of time and say, look, what have you done? You've said you were going to do this. You took part in what is this popular movement over the summer as a result of this awful event. Now what? What are you doing about it? And also those organizations that said stuff without any commitment, they also need to be brought into this sort of conversation and be like, okay, you've said something, or retweeted something or something like that. What have you figured out what's wrong with you yet? Because every organization within science and health research, for example, there is racism entrenched in the systems, in the policies, and in the processes. Diversity as an output is like a symptom of that. And you can report on diversity statistics and say we're not diverse enough, so we need to do something about it. It's like, okay, so what is the ingrained systemic racism that is really driving that, and how are you going to reflect on that as yourselves, as an organization and as individuals, to get rid of that and to change it? And that could be destroying that system and creating a new one, because it has been designed by white people, basically, for essentially white supremacy. That's something that is a hard conversation to have, but needs to happen. And I think that's starting, like, making sure that organizations are doing the hard work around that, I think is really important in this moment as well. So that, in terms of racism and anti-racist learning and improvements, plus global pandemic and the stresses and general inequality that's been seen as well around other areas too. I think the fact that those two things have happened at the same time as well, like, we really need to latch onto this, and we need to do something, and we have the momentum, I think, to do it. And I think, it's exciting. It's scary, because it always is. Burning something down and starting again will always be scary. And I'm not saying that's what we're going to do necessarily. This is no criminal damage going on here, but it is very much like okay, this feels different. This feels different this time round.
Interesting. You're talking about sort of not making firm commitments. It's interesting. I'm sort of in a position, for one thing, where I wanted to say this is bad, but I didn't want to be making any promises about what we were going to do about it, because I don't know what the issues are, and I can't promise that I'm going to change things or make a difference until I know what that is. And I think, like, a month later, someone's like, so what have you done? And it's like, well, we've not seen anyone, we've not been able to talk to anyone, everyone's away. Like, I don't want to be over promising and under delivering. At the same time, you want to make it clear that you're genuine about it, right?
100%. Yeah, I think it's the biggest thing. So every organization and every individual was at a different point in their own journey around understanding racism, systemic racism, and developing an anti-racist approach. The question is, if your organization or anything like that hadn't thought about this before, this would have been a wake-up call, going, oh crap, we've not thought about this before, and this is big. This is a big thing. And that first section there can include fear as to, we've no idea what to do or where to go. Now, thankfully, because so many organizations were either having these wake-up moments or these public moments at the same time, there is a lot of information out there. There always was on Google, but it's so much more obvious in the public sort of eye and in the sector as well, that there are places to go to find information out, organizations who have put their own statements, their own action plans out there already. But what probably will happen, without committing to, we're going to change all of our ways because we don't know which ways need to change, is that first sort of self-reflection of going, okay, we need to look internally at our own organization and really figure out where racism plays a part. Where have we allowed this to happen? Where have we enabled this to happen even? And start there, because that's something. And that could be, for example, looking at the symptomatic, diversity data, seeing where have we got clear underrepresentation. Could be the entire organization. It could be, very specific levels of seniority within the organization. Could then be looking at the policies and the practices and going, okay, are we gatekeeping in some way? Is that gatekeeping related to other social inequality, including racism? Is that something that we can look at? And it's really sort of like taking that time to self-reflect and figure out what is it, where is it, and how have we enabled racist structures to occur within our organization and therefore within the sector.
Yeah, I was in a meeting last week. Time is a weird concept right now, but I was in a meeting last week and I said, "Where's the data for LGBTI plus people?" So the other diversity data was pulled out, and that wasn't. And they said, "Oh, well, the numbers are too small, so we haven't got data." And I'm like, "Well, the general consensus is that LGBTI plus people make up 7% of the population. So if you've got data that's too small, that means that you're not in target." And when we're looking at schools now, we're talking up to 45%. So if you've not got any data, and it's the same, I think, with race in certain positions as well, the data's too small. It's like, well, yeah, but the population isn't. Why don't the population and the data that you've got match? Where's the problem? And 100%. They were like, oh.
Yeah, if you can't report the data because the percentages are too small, for example, you already know that you've got underrepresentation. You can't then not comment on it. Missing data is still important to know, because you still know if you've got if people, for example, have been asked to voluntarily input diversity data, for example, and you find out that most questions are answered 90% of the time, but the one on sexual orientation or the one on gender identity is answered 50% of the time, there's something wrong there. Not in terms of like you can say what proportion of people you're representing, but you can say you have an issue with how people feel about confiding this data. Therefore, people are concerned as to how this data reflects on them and how this data might be used on them, which more often than not says, I don't feel comfortable telling you, for example, like that I'm queer because I think you're going to treat me differently as a result of it, or there's something like that around it. So when it comes to data collection, there's diversity data collection, there is so much important information to be had in the way the questions are asked, the way they're answered, and then also the results. It's not just here's your numbers at the end of it. There's more to it than that.
Yeah, I mean, I think the meeting that I was in last week, I think I was a little dismissive because they are on it and they do appreciate this. I think they just didn't realise that, like say we're talking 7% of the population upwards is LGBTI plus, and I guess that's all about which circles you move in, who you have around you, who you're friends with, and our circles are our circles. It's as simple as that. And sometimes I know in Oxford, it's a strange place, so I can understand how that happens. But that's why I think we saw a letter that was published, not in a scientific journal, but in another journal by scientists saying that there isn't a problem with racism. And it's like, well, that's because of who you're talking to. And like you say, sometimes the absence of data can tell you just as much as the actual data itself, I think.
Maybe. And that's why we talk about, like, when we talk about groups and we talk about diversity, the reason a lot of people say, like you get a diverse group and that's the solution sort of thing. And it's like, actually, it's the process itself. Like, are you talking to people with different lived experiences, different backgrounds, and therefore different perspectives to bring to the table? The other thing is also thinking about how people prioritize. This is something I'm really stuck on at the moment, which is like everyone prioritizes stuff based on their experiences. So therefore, pretty much everything we do in work, in research, in everything, we're creating priorities. We're deciding what's important and what's less important. And the only way that we can do that in a way that is fair is to, one, amplify the voices of those from minority groups who aren't able to have the same number of voices at a table, for example, but then also to bring those people into that conversation and make sure that they are involved from the off, so that we are getting everyone's perspectives from that first point. If you were to say, think about, what's people's favorite color, for example. This is like a completely sort of side of thing. If you were to say, what's the UK's favorite color, and you go out and you survey a group of people, and you survey a group of people who are all sort of the same in some way, who all, a group of Chelsea supporters, for example, they're all going to say blue. But that doesn't mean that's the favourite colour of the whole of the UK, nor does that have any granularity in terms of the other favourite colours. So you can't go around painting all the streets blue and thinking that everyone's going to think it's great, because it doesn't work like that. You need to bring other people into the room. And also, if you've got someone out there who is allergic to blue, for example, you need to make sure their voice is being heard, so you don't go off and paint everything blue and then they have an issue. Like it's like the weirdest sort of like comparator in the world, but it's the way that I'm trying to explain it in a sort of like this abstract manner of why it's so important to have everyone's opinions around the table, and why it's so important where, especially if you're in a group of people, you think about who has the loudest voice. The loudest voice will be the majority group, because it'll be the amplification of the same voice 10 times. So therefore, you need to find other ways to amplify people's voices who are in the minority groups, whatever that is, because otherwise, no matter how much you try to have everyone at the table, their voice will still not be heard as much as the others if you just have this sort of like equal version of things. You need equity. You need to be able to amplify the voices of those from minority groups, and you need to be able to make it fair in terms of how they're heard and who's being heard in decision making, because otherwise, it's this idea of like, okay, let's say we get a representative of the population around a table and everyone has a single vote, which sounds really fair. You've still got a majority. If you're voting in your own personal interests, you still have a majority, therefore having complete control.
Yeah.
And it's like, it's suddenly you're like, oh wow, okay, so do we need to come up with like a different version of democracy within decision making in order to make sure that we're doing this actually equitably and actually fairly? Because otherwise, those who are in minority groups will always not be able to have votes that are equal, because they're still a minority. So therefore, you need to start, one, increasing number of people from minority backgrounds into that decision making process, and two, making those who are in the majority groups not vote in their self-interest anymore, but vote in the group interest. And it's those two things together that are so important.
Yeah.
Got me on a tangent now.
No, but it's a good tangent to have. And it's interesting when I'm thinking about it, because I completely agree. Like I said, there's not enough LGBTI plus people in science, so we need to increase it. But then, I don't know. I'm starting to have complicated opposing thoughts as well, though. Like, how do you? Because occasionally you'll have extremist views, and those extremist views are just there to be contrary, or due to a lack of, due to ignorance, or whatever it is. And I was talking about those academics who are writing and saying racism isn't a problem and women are less intelligent than men, was another paper. And so how do we balance those views?
Yeah, I mean, this is the thing. You got to remember that underlying this is you've got this idea of intent and this idea of social justice. So firstly, the intent has to be good. The intent has got to be, you're trying to do good things. So these people who are out there playing devil's advocate because they've got no investment in the game, that shouldn't be allowed. You shouldn't be able to just be like, oh, I'm doing devil's advocate for the sake of it. That proves to me, one, that you're privileged enough to not feel like this is a personal attack, or that this is going to actually affect you if you feel like you can pose these opposing views for no reason. And two, is it actually going to help the conversation or move on at all? Probably not. Thirdly, you need to think about, like, okay, how are we doing this in a way that's not harmful to people? You've still got to have people's genuine human rights at the core of this, of the core of all of the equality work that we do. It's about making people's lives better as well. You're doing this because it's fair and it's the right thing to do, and it is just. It's not trying to diminish a group. It's not trying to cause harm to someone. It's when people pull out things like, for example, like you were saying about sexism around like intelligence and things like that. Actually, firstly, it's not based on real science, not based on true science, and it's just sort of like, okay, firstly, you need to make sure that you've got the evidence behind that. If you're going to start making these claims, it's not true. Same thing as well, like saying there isn't racism. What's that benefiting? What's the benefit of writing that so that people don't do anti-racist work? Anti-racist work is important and it is good. We need to do it. Racism does exist. Denying it is, denying racism is like climate denial, okay, climate change denial. There's stuff that out there that we know exists. We're scientists, so we have evidence anyway. But like, it's not productive to deny these things. So when you think about like extreme views and things like that, are they doing harm? Are these harmful views? That's the difference. It's a harmful view. It's going to cause harm to a person and to a group of people. That's different from someone's voice not being heard because they're being discriminated against. And I think that's something that we have to remember to weigh up. I think when it comes to, for example, thinking about like LGBTI inclusion, again, it will likely be that LGBTI people are in the minority in pretty much every single sort of decision-making setting at the moment, and so it's like, okay, so when someone says something like, in terms of like healthcare, for example, someone says something about healthcare and we make a decision, and the decision being made at the moment, is very heteronormative. If someone puts their hand up and says, look, have you thought about this, this version of events that is actually inclusive, therefore, of LGBTI people, and that person might be LGBTI themselves, which is why they've noticed it. Just because it's one voice doesn't mean you dismiss it. You go, actually, that will benefit this group. And also, when you design processes or systems for some of the most discriminated against or most marginalized people, often you'll create a process that's actually beneficial to everyone else as well. You can design something, for example, design the entrance of a building. You put a ramp there instead of stairs there. Everyone who could use the stairs can still use the ramp. More people can now get in the building because the ramp is more useful. Oh, and it just so happens that I, who can use the stairs, can benefit from the ramp when I want to bring a suitcase in. There's all these additional benefits that happen when you design processes and systems to really support those who have not had their thoughts and their needs designed for previously. Yeah.
Yeah. No, I mean, this is it. Like, true progress benefits everyone. It does. And this is why, for me, I've started branching away from just sort of LGBTI plus equality and worked into more sort of like making STEM, like yourself, STEM more inclusive and equitable for everyone. Because all those changes benefit everyone.
I mean, a clear example. So on Monday, we've got F1000 Research launching a new policy for name changes in publishing, which I know is something that has been mentioned as particularly difficult to do.
Yes.
That we've got trans researchers wanting to change their name on previous publications so that they have more ownership of those papers. The processes and systems are not transparent. They're not even necessarily done by a lot of journals. So we've worked with F1000 Research to come up with a policy that works. We've worked with researchers to review this policy and check it works for them, and it's done in the right way, done in a respectful way, done in an empathetic way, all of this sort of stuff. And when we realize, this policy actually is going to benefit people who want to change their name as a result of marriage, of divorce, especially, considering if it's divorce as a result of, I don't know, domestic abuse, and then, a previous name brings up also incredibly negative emotions as well. If they want to change their name as a result of religious conversion, the policy has been designed in a way that is so inclusive that all of those groups will also benefit as well. And it's just such a neat way of designing things where you just consider, okay, what is the hardest version of this for someone at the moment, and how do we make that right? And then you will sort of bring up all of the other people as well at the same time.
Yeah, it's weird. It's one of those things, like, I never want to cite all papers or share all papers because I don't want to see my dead name. And also, I've had this, this question's come up a few times of like, do you disclose that you're LGBT? And it's like, say in job interviews, and it's like, well, for me, I don't have a choice because it's there on my CV. I've got a different name. It's obvious. I mean, so it's something that I know me and other trans activists have been talking about for a long time. So it really is great. But Yeah, I mean, we were starting to talk about, well, what about the victims of abuse? What about people that need to get away from other lives or whatever it is? There's not just not just trans people that benefit from this. So it's
No,
really great to
exactly.
hear. It's
Yeah, and
awesome.
it's been really great to work on that. Firstly, F1000 research and Welcome Open research have been really responsive in terms of sort of how to do this as well. So that, they came to us and they're like, we've come up with a policy, what do you think about this? I'm like, yes, we'll go through it, but we need to involve researchers who are going to be affected by this, who want this policy change, for gender recognition, but also, just making sure that we've got this covering, you know, everything that's correct about this. Because, I'm not a trans researcher. I can't comment on whether or not it would be the right process for me. So it's making sure you bring those voices in. So they were really good at like then slowing down to make sure that we got that feedback. We had 3 researchers review the policy, review the blog post that's going out and everything around it as well, and make sure they were happy with it too. And because even within that group we had diversity of other characteristics, we were able to pull out other things that we also wouldn't have noticed. So for example, I didn't even clock that there would be gendered surnames existing, but they do exist in other countries. And that also needed to be made sure it was addressed when it came to, providing evidence for this as well. What was the best way of doing that? That meant that someone didn't have to go through another process which had its own barriers to do this and things like that. So I think it's a really good example, and it also shows, it was a good, I think, demonstration of the fact that to do this well takes time, and you have to admit that, and you have to do that, and you have to put the time and energy and effort into it. But by doing it well, it means you're not going to have to redo it, and that's also really important.
That's the thing. It's doing it properly. Although we've still got to realise that, we've got to review these things every couple of years,
Yeah,
no matter how well you do it, because things change.
of course.
I think you're in a really unique and interesting perspective. The other people that I've been talking about, we're doing the sort of diversity and equity stuff on the side, as it were. Whereas, I mean, this is your sort of bread and butter. This is what you're doing at the moment. I mean, is that a fair assumption? Is that right?
Yeah, no, absolutely. I think it's the one thing that I've really enjoyed doing, and I think it's the kind of path that I've forged a lot, is that the diversity, inclusion, and equality and equity work was obviously a passion of mine. I'm a queer woman that was working in STEM. There was a certain amount of like drive for me to set up the LGBT plus network at the Crick when I was there, which in part was selfish reasons. I wanted support and I wanted to offer that to other people as well. And it was like a reflection of my own time going, it would have been great if this had existed. So therefore I want to create that. But the other thing is also then being able to merge the research with it as well. So, my own perspective in terms of like the inclusion of actual research design has hugely changed over the past few years in terms of like understanding more and learning more about it, and sort of really understanding sort of how you can embed inclusion into the system, both in terms of who's doing the research, how the research is done, and then who benefits from it. And it all works as one giant sort of like machine that with all the cogs turning in different directions. But the whole thing's really important. So that's been really nice to be able to pair my research experience and my actual research topic work in terms of thinking about like population genetics with social justice and equity work as well, and merge those together into like an actual full-time job at the moment, and that's been really great to do. It's also been really interesting to then get, to kind of move some of those thoughts that were really ingrained into my brain as a result of like the research side of things, and sort of like push them out so that the social sciences could kind of move in a little bit further, because there's loads of stuff that like, wasn't included in my undergrad or wasn't thought about, when it comes to, especially with genetics, really understanding stigmas associated with genetics research in different cultures, for example. That should have been a topic really in my undergrad, but it wasn't, because we were focused on the technical aspects so much more. So being able to sort of merge all of that together and give myself sort of a refreshed view of a lot of the research that I have done and that people are doing now has been quite great. And it has been exhausting doing EDI work as a full-time thing, because something that, you used to be able to separate somewhat my personal identity with the work that I was doing. Often, my personal identity could be something that I could actually leave at the door if I really wanted to. If I wanted to just get my head stuck into genetics and like sit at a lab bench and do that, whereas it's impossible to do that with the work that I do at the moment. It's impossible to not feel a personal attachment to stuff that we talk about, stuff that we're doing, but also stuff that affects people as well. And it is very much that idea of like everything that we do is going to affect people, and people are real. And people, people need to be thought about, and their emotions are valid. And sometimes we'll get it right, and sometimes we might get it wrong. And that's also something we have to, be honest about. And then you do that work sort of in your day-to-day life, and then suddenly, things happen in the outside world that are also completely relevant. Thinking about like the Gender Recognition Act, the changes that have been, and the voices that have been around that sort of over the summer, especially like on social media, like people being for and against it, and that side of things. And then people outside of work therefore looking to you to put input or to talk about it or to educate and things like that. Even for example, around the Black Lives Matter movement upsurgence, people then talking about that outside of work, and everything just sort of like merges through into this constant thing. Add on to that working from home, and it's like, cool, been on 24 hours a day for 6 months. Like I'm like on call having conversations, like even tough conversations with like friends and family members, trying to explain why it's okay to say Black Lives Matter, and it's really not okay to say all lives matter, especially at the current time. Like having those conversations, you're doing that and trying to do that kind of outside of work, and then also sort of like firefighting stuff like within work as well, to make sure that the same thought process and the same care is being taken to people's words and actions. It does. I can see why a lot of D&I practitioners end up having some form of like burnout, in the same way like researchers can have burnout. When your work becomes your life, it really it takes you down a certain path. I guess it's that idea of like, how do you harness that passion in a sustainable way? And that's really something to talk about, and that's related to both my work within EDI, within science and health research, but also within researchers themselves. And I see it happening so often. There's an expectation that you're going to be rolling over these short-term contracts, which are like constant sprints of research and constant sprints of productivity. It's not sustainable. People can't do that forever. And the people that can do that forever, but they're often the ones that can do it because they've got other luxuries in place, other privileges that have allowed them to do that. That they're not fighting homelessness at the same time. They're not fighting, continuous microaggressions at the same time. It's that idea of resilience to that sort of process and that system. And again, why are we asking people to be more resilient to the system? Why can't we just change that system?
It's interesting. A lot of what you've just said sort of quite resonates, because just yesterday I saw a job come up in my home city of Manchester, which is an EDI job, and it's a permanent job, as much as any job is permanent. Whereas my research contract is coming to an end my current contract is coming to an end in a few months, and so this idea of having the luxury of a permanent job is great. But then again, I think maybe I'm too ingrained in the kind of activism, and also I won't be able to shut my mouth. I would take things personally, but the point is that I can also walk away when I need to. At the moment, it's like, no, I'm doing this in my spare time, and so I can turn off my phone for a week and not get involved in these things. And I'm fighting to change things, but I don't necessarily have responsibility either, and so I've been thinking about it a lot, that whole thing of like permanent job in my home city, where I might actually be able to save some money rather than in Oxford, where yeah. But someone I talked to a few years ago, I was looking at EDI jobs, and they said it's a golden birdcage. You might get paid for it, but you won't be able to do what you actually want to do, so you might not. I don't think it's for you.
Well, that's the thing. It's that whole sort of like understanding that, yes, I love my work and I love what I do, and I love working with the organisations I work with, and seeing change is always amazing. And also sowing seeds for future change is also still something that's particularly good, and something I have to remind myself, like, okay, might not see the immediate change, but there is going to be something coming up. That's always great. But yeah, acknowledging that you are still working within this system, which has its limits, is quite tough. And sometimes you butt up against this sort of like processes that just mean that you can't do the thing that you want to do, or the thing that you know you should do as well. And that can be quite tough, and it can, I hear people say it's a thankless job. It's not. People say thanks, and it's really lovely when they do, and I really appreciate it. So if I've ever helped anyone, say thank you. It makes my day. But like, it's just making sure that like there's an emotional resilience that needs to happen, and I think that's kind of tough to kind of explain. Like, I wouldn't have been able to explain that to myself 3 years ago before I got into it full time, that I would need to have that, and I'd need to know when to be able to switch that on, and be able to do that side of things. I will say it's quite nice being with EDIS. Technically we're hosted by Wellcome, but because we're sort of this half in, half out, and we're this coalition of organizations, there's a certain amount of protection that comes with that sort of mass of organizations, and then having the own EDIS identity, that allows us to be a bit more experimental in the stuff that we talk about and the stuff that we want to do, and sort of push some boundaries in different places that we wouldn't normally be able to sort of push necessarily. And that's quite nice to be able to do that. So I think that's the difference, where I've got that sort of escape from working without working in a really strict system. That it's that little sort of extra bit of freedom that really works. But yeah, I think the idea of being able to sort of like switch off when you need to, or walk away from stuff, that's tough, and that's not something that can happen. And that's something that I think everyone that I spoke to who works in D&I, both in Wellcome but also in other organizations as well, talks about how do we prioritize what's going to change, or what needs to change, what projects and things like that, whenever someone comes to us asking for something to change, it's because it's not right, and it's because it does need to change. So saying no is really hard, because in your heart of hearts that does need to happen, but there's only so many people, there's only so much money and things like that. It's like that's the tough, because I think everyone I speak to who works in D&I cares. They care about it. They care about it for other people. They care about making the system better. They care about being part of equitable and fair society and social justice. So it's like, of course people are going to be passionate about it, and of course people are going to put their all into it. And I see some of my colleagues doing incredible things, and I see the amount of work and energy they put into it because they care about other people, basically, and it's really nice to see. It's just a case of making sure that can be sustained, and that is sustainable, because you don't want people burning out, and you don't want people giving up either. And it's just making sure that works, which I think is also why organizational culture is really important. Make sure that you support people who really care, who put a lot of energy and effort into it, who make sure they're rewarded well, and make sure they're supported in like other areas as well.
Yeah, great. We need to support this. It's simple as that. Everyone needs to support it.
And I think this whole sort of stuff where I talk about this in terms of like people working in D&I is also so relevant to people working in research, because so many people become researchers because they're passionate about a topic, or creating something for the benefit of society, or like finding a cure for something. All these sort of things are passionate things that people care about. They do just pour their own heart and soul into that research and into making those things happen, and then get tied up, by having to then jump through a million hoops to make sure that you've got continuous funding or contracts and things like that. And it's like, if you're wanting someone to really to work hard because they want to, and to the ability that works for them and sustainably for them, working sort of in the best way for them, something they care about, you need to make the rest of that system easier for them. You do not need to put them on precarious short-term contracts. You do not need to whip their funding out from under their feet at a moment's notice. You do not need them to funnel all of their research outputs into a very narrow set of criteria of what success looks like. You need to allow people to work in a way that works for them, and a way that allows them to harness that passion in a sustainable way that would then benefit society. And that's something we're not doing right at the moment, that's for sure.
Yeah, I am sort of advocating for kind of like, I guess, the UKRI Future Leaders projects, where you're funding a person rather than a project with outcomes. You're funding that person build a career, basically. And I was encouraging other people to do that. The UKRI doesn't get everything right, but you know, I think that's a really good project. And yeah, admittedly, there was a little bit of selfishness at the group I was saying, encouraging that they do something similar. I was like, and give me one, but you know.
But this is the thing, though. This is there needs to be a wealth of solutions, because no person is the same. This is something we always try and come up with a neat solution that will help, or that will solve all the answers. Reality is we're going to need 10 different versions of funding streams, so people can pick the one that works for them. Some people want short-term contracts because they want to move around loads. Some people really don't, because they want to stay in one place. People will want different things, and there needs to be options available for everyone, and in a supportive environment as well. And I think, this idea of just streamlining everything into a one option just doesn't work.
I agree, and even silly stuff like wages. I mean, it costs a lot more to live in Oxford or London and other cities than it does to live in others. I remember there was an EU project that I was part of, and they used to give out money for short-term scientific missions. And someone got one to come and work in my lab in Switzerland, realizing that Switzerland's not EU, but we were an associate. And the money that they gave for living costs didn't cover half the cost of a hotel per night in Zurich. But there was no leeway on that. So if you went to a country where living was really, really cheap, you went home with a lot of money in your pocket. And if you went to somewhere like Switzerland, you'd
be out of pocket.
Yeah, you were out of pocket. They ended up staying in my spare room.
Then that opportunity is only available to those who can afford to be out of pocket, which already, exactly. Where's bam? That's a barrier that was right away. Think about think about when that money gets put into someone's pocket. Think about students and how often people would have to pay up front for travel and then get reimbursed as a grant. And I'm like, I'm sorry, I did not have spare cash for travel or conference fees, ever. And I was supposed to cough that up and then get that back next month.
Well, I went to a conference last year, and it took me 7 or 8 months to get the money back, and that was flights, hotels, and Oxford's expensive. I've got no spare cash. It took me a long time.
Exactly, that's the thing. If you're choosing your career path and you know that that's something that you might have to do, and you don't have that backup and you don't have support in other ways, you just can't do it. That door is closed. You can't go to those conferences. You can't do that traveling, and therefore you don't get to have the same opportunities to advance your career that other people have. This is the thing. These are the things. This is a discriminative policy is in place. This is systematic discrimination of people who don't have financial security. That's a policy that people say, oh, it's you reimburse the money. Discriminatory. So you need to change that policy. This stuff like this is the stuff that needs to change within the system.
Yeah, I grew up on a council estate in North Manchester. I don't come from a family with money. It's working class background, and I like that they're trying to push for people from different socioeconomic backgrounds to come to Oxford. But you can't just sort of give them the space, because it's too expensive to live here. Even if your accommodation's sorted, just living here is a lot. And so it isn't just about recruiting from those schools, because they're not going to thrive or succeed if their mates are going to balls where they pay 200 quid for a ticket and they can't even afford to get the outfit that they need to go in. It's not the same. Very passionate about class and social economic background, but it's something that I don't talk about as much because of the trans thing.
It's frustrating, because it's one of those ones that it's actually socioeconomic background is quite an important one, and it has a lot of intersections with other aspects of diversity as well. It's also important. You do need to consider it, be it not a protected characteristic. I think it is really important, because let's say we've got a diverse group of people, whatever that means, making a decision, but they all went to Eton. Oh, am I talking about the government right now? It's not the same as actual diversity of experience as well. You have to make sure you understand those intersections between different aspects in order to do that. Albeit my background, my growing up was slightly different. I do remember you know, payday happening for my parents and being able to get a packet of munchies as a treat rather than a 10p Freddo. You know, like there's, there's certain aspects of like things that happened where you look back and go, oh yeah, that was quite tough. And like having to work for a year and in order to make sure I could afford to actually go to London for my undergrad and working every single summer, you know, I look back at my Facebook memories and I seem to be complaining about 17 days in a row of cafe work, you know, every single summer. You know, scraping together, like this is the other thing, like mobility around geographic mobility as a result of socioeconomic status and background. You know, you can't just move to a new city unless you've got a deposit for like rent or something like that. You know, you for travel, for hiring something to take your stuff there. You know, I remember going down on the train like 2 suitcases, and that was how I could get my move into London. Like that was what I could do because I could get a train fare and I could get my 2 suitcases, but driving like a car or a van to move my stuff, like not feasible at that point, you know? It's just, and so when people talk about this stuff, they're like, oh, you know, once you get here, you'll get your first like stipend, for example, for, you know, your PhD, you get that at the end of the month. I'm like, I've got to wait 4 weeks until I get money. How, how am I supposed to survive? You know, like there's no backup funds because of where you come from. You know, it's yeah, the things that they get put in place where people are like, oh, that's a good thing. You're like, is it though? Is it really?
I have lots of views on this and renting and things like that. And you were talking about deposits. I mean, you know, I could move, which would save me maybe £100 a month in rent, which would make a huge difference. But I don't have the money to do that because you've got to pay for someone to move all my furniture. I've got to have that deposit up front, and I'm not in that situation right now. Like, it costs money to save money. And yeah.
I mean, I remember pawning some family jewelry to make sure we got a deposit for a flat whilst at undergrad. You know, like there's certain things that, you know, people just don't expect you to have done, and you're like, well, that's how you get cash when you don't have cash. You know, like it's people just don't think about this sort of stuff of like the amount of like buffer you need to have to do an undergrad, to do a PhD, you know, to go to a different university for a postdoc, stuff like that. Like you need buffer money, and that just, if you come from a background where that's not a thing that you've ever grown up with, how do you find that?
Yeah. Yes, I'm thinking heavily about it at the moment, shall we say. The cost of living in Oxford versus living in Oxford. I love it here, but holy moly. That's all I can say. Especially, and you pay a single tax. If you're single and too old and grumpy to live with other people, you pay a premium. It's simple as that.
Yeah.
And I am too old and grumpy, I've found out, to live with other people. Turns out, not so good at it. Cool. Well, I realise that we've talked way longer than an hour, and so
wow.
but it's gone so quickly and so easily. Is there anything else that you did want to mention? Anything else that we hadn't touched on? If there's not, that's absolutely fine, of course.
I'm trying to like, what have I talked about, and what do I want you to cut out of this conversation? No, I think
that
we're good.
Can also be done.
I don't think there's anything particularly else that I want to add on. I mean, I always what's it called, give a poke for people to join EDIS if they're part of an organisation that's committed to improving EDI and want to work without, you know, leave egos at the door and come and help the whole sector as a whole. I think that's my biggest push for EDIS. I think they'll leave on that.
EDIS is MedSci.
So yeah, science and health research. So really thinking about so our impact statement is equal and better health outcomes. So anyone working in sort of any organisation within sort of biomedical health research type fields, medical research type fields, but also any organisation that's involved in advancing the EDI agenda within science and health research as well. Really interested in hearing from other organisations. You know, there's lots of grassroots organisations that I want to make sure I have access to, the organisations that are part of EDIS too. So always happy to connect everyone up as much as possible.
Awesome. Well, with that, thank you so much. Thank you for having this amazing conversation. And it's really nice to see you again, because it turns out it's been quite a while since we've been able to see people, and so I haven't seen you for quite a long time. So, and we're always busy when I see you as well.
Yeah, one of us is either talking or like running around after someone. So yeah, it's always love to speak to you. Thank you so much.
Usually talking. Cool. Well, thank you so much. Bye bye. Hello again. So that was cool. I really enjoyed that. We were talking for ages. I'm so pleased with when I was chatting with Chris the other week. I mismanaged the timing and so didn't get to chat about everything we could. This time we still could have talked for longer, but it was good to have a really in-depth and long conversation about Lily's work and what she sees in the world around in STEM. So that was really exciting. I really enjoyed it. Like I say, if you've not watched our previous ones, please do go back and watch them. I know they're quite long, but I think they're kind of cool. And we've also started releasing them as podcasts. So if you look for, I think it's Science Chats with Dr. Clara Barker. You think I'd know that better, wouldn't you? But it's available on sort of Spotify and Apple and other podcasty places. And if there's one that you know of that I haven't put it on, then let me know, really. Yeah, so if you don't want to listen to this, like I say, on YouTube, watch it there. So yeah, thanks so much for watching. If you can like, subscribe, and all that malarkey, it's always good. It helps. I'm trying to get a bit of momentum behind these, so please do share. It does help a small YouTuber like me. And yeah, next time in a few weeks, I'm going to be talking to Bernardi, who works for a science publication company. That was a fantastic conversation. I just did it earlier in the week at the time of sort of recording this, and it'll be out in a few. So that was a really good, fun conversation. And I'm looking forward to having more awesome conversations with basically amazing people who are scientists, and also really fun and just great to chat to. Scientists, with people, and it's nice to hopefully bring them in. With that, take care, and I'll see you again soon.